I should start by just thanking Dr. Litvak, Dr. Kops for the opportunity. And for many of us, we come to these meetings, we fly in, fly out. But it's a heavy lift getting this thing to work and run on time the way they are. So I know you put a lot of work into this. It's evident. Congratulations on the course. I'm always surprised as I see different introductions to brain tumor talks and how people are going to frame things. I was recently at a talk from a kind of a well-known chair of neurosurgery, and you can spin the data how you want. There are ways and data sets to show improved tails of long-term survival. And then there's this data, which is really the amalgamation of all of the national data for survival, which really tells you the story that we haven't really made much impact as much as we would like for certain. But we have to be honest with ourselves when we're trying to look at the problem we're trying to tackle here. This is a difficult problem. Some of the people in this room and many others around the US and the world have contributed to what is currently the standard of care for these patients. Surgical techniques have improved vastly. We are much better than we were even 5 or 10 years ago at removing these tumors. Radiotherapy, which we haven't heard much about today, has accelerated incredibly. Our ability to deliver safely high doses is exceptional. We typically have one drug that we rely upon for these patients, and that's sort of the focus of this talk. And more recently, we have Optune, the Tumor Treating Fields, which is a proven technique with level 1 evidence to support it, and has given us a fourth road forward in treating patients. But all of this together still leads us to the survival curves I just showed you. The real disappointment in all of this is what hasn't been delivered at the systemic therapy level. And as everyone has alluded to today, this is not a local disease, and so the solutions are not going to be local. In the past 30, 40 years, we've seen 5 drugs approved in the US with a malignant brain tumor indication, most recently in 2018 with everolimus for patients with tuberous sclerosis and subependymal giant cell astrocytomas. It's a very, very small subset of patients. So we really have Temodar, which is unfortunate. If you take a step back and you ask yourselves why this is the case, it's not for lack of brain power and it's not for lack of trying. We have a system in place similar to in Europe where we have to go through these iterative steps. The challenge is that these iterative steps are really a lot more difficult year after year than they used to be, a lot longer, a lot more fraught with risk, and importantly, a lot more costly. And I think this is where the narrative sometimes goes astray from our reality. 20, 30, 40 years ago, new drugs and new drug concepts came from academic medical centers like Swedish, like UCSF, like the Barrow, in university settings. That's really no longer the case. New drugs are developed by industry today. The agenda for new drugs is developed by industry today. The indications for new drugs are designated by industry today. And for good reason. The non-capitalized cost for just pursuing a single drug is about $500 million. If you throw in all the capital costs, is around $1.4 billion. And that's the conservative side of the most recent estimate that came out a couple years ago. If you look at the bottom graph, that shows you where new drugs are actually coming from in the last 20 years. The lion's share are coming from pharma and biotech. And if you look within those industries and ask yourself where they're allocating their efforts and where they're developing their successes, what you really need to look at to answer that question is where the market sizes are. Brain tumors are not a billion-dollar market. And as a consequence, new drugs developed for brain tumors are exactly proportional to new drugs developed for lung and breast if you factor in the market size. Now, this is not to indict the industry. In fact, they're doing exactly what they should be doing. This is how the system is set up. But we have to understand these rules and realities if we're going to actually change the performance that we've had over the last 30 years. At the Barrow, we've recently initiated another Ivy Center. This one, we've tried to build with all of these realities in mind. When you typically build a research program at any university, and all of us can harken back to this from our training, usually what you do is you pursue the best and the brightest, right? And you bring them together, and you try to give them an optimal environment. The challenge with that system is that as much as you would like to coordinate these recruitments and establishments of efforts, the fact is that most of these efforts are really parallel tracks. Hopefully they intersect, but at many centers, a constellation of laboratories is really a gladiator academy, where some of them will succeed, some of them will not, some of them will work together, and some of them won't. At our program, we've been fortunate enough to really build something where we've kind of removed that. The laboratories that we've pieced together and put together specifically for this program function more like cores, where the investigators, scientists, and everyone else really have a mandate to pursue certain activities as part of a greater blueprint. So it's not about an individual investigator just pursuing whatever makes sense to them, either scientifically, financially, or otherwise. It's really about everyone specifically pushing in a coordinated direction, and that's how they're recruited. So there's no surprises when they arrive, and we ask them to go in this particular direction. Because we have about 1,300 brain tumor operations per year, this gives us a special advantage in terms of tissue-based trialing, and that's what phase 0 trials are really about. But if you talk to anyone that does clinical trials, what they'll quickly admit, and it's common knowledge, is that the toughest parts about trials are not the scientific parts. It's the administrative logistical parts. It's the fact that if any of us today have a concept for a clinical trial, the first thing we're going to do is we're going to wait 12 to 18 months for the lawyers to get back to us to let us know how we can proceed with this. And this is just reality in 2019 in our regulatory environment. In order to sidestep this, we built this center within a separate nonprofit LLC. It's actually independent from the Barrow itself, from the academic medical center that the Barrow resides in, and from the master healthcare plan that owns that academic medical center. So what this means is that all of the parts and pieces of a clinical trials infrastructure answer to our center, are funded by our center, and therefore everything from the regulatory specialists to the lawyers and special counsel that actually drive our trials infrastructure are aligned in their interests and in their priorities. So it gives us a level of efficiency that basically takes most clinical trials for us a couple months to get up to speed and actually start accruing, and hopefully we finish accruing within a couple months as well. So we're trying to do everything within calendar years. So let's talk a little bit about if you have kind of an optimal environment for drug development, how you develop it. Well, you know, the barriers to drug development, the reasons that we haven't been successful are pretty well known, and some of them have been touched on today. Penetration, CNS drug delivery, major problem, and oftentimes overlooked as we fast-track drugs into phase 2, 3 trials. Most of the drug development efforts today are based upon preclinical data. Preclinical animal models are very useful in some respects, but they can be misleading in others. We don't have animal models that actually recapitulate what happens in humans. We have plenty of therapies that have positive effects on the animal models and do the opposite in patients. We've heard this several times today, the need for combinatorial therapeutics. I think everyone recognizes that intuitively, but if you look at the literature out there today, just on your computer as you're reading them week by week, these are monotherapy studies by and large. When we do combinatorial studies, it's often a new drug combined with an old drug, and that's really probably not the right direction for us, certainly if you want to be ambitious. The phase 0 trial concept is something that was introduced over a decade ago. It was developed by the FDA, not for neuro-oncology, but for medical oncology. And the idea behind it was that they wanted to have an ability to do quick trials with drugs that were early in their development, where you can just quickly get a sense of whether this drug had any promise or not. So the term they used when they introduced the concept was humanizing preclinical studies, meaning asking the questions you would ask in an animal model in patients. These were typically small studies. They weren't necessarily first-in-human studies, but they were early-phase studies, 10 or 20 patients, using a concept called microdosing. Microdosing means for systemic therapy, like if you're developing a drug for melanoma, you're giving the patient 1/60th of the therapeutically active dose. So basically no risk to the patient, but obviously no therapeutic benefit either. The idea being that you would give them this dose, you would get a sample of their tissue. So if they have lung cancer, you would do a needle biopsy on Monday, give them a microdose on Tuesday, and on Wednesday do a needle biopsy again, and basically ask two questions. Did the drug get there, and did it modulate its target? The problem with that approach, and the reason why it hasn't really been embraced by neuro-oncology, is because that system doesn't really work when you have, for example, a blood-brain barrier. If you give a microdose of any drug, including Temodar, it will not get through. So we've had to make some adaptations to this model. The first is by changing the dosing strategy to something higher. Still not high enough that there's meaningful risk, but high enough that there's actually a chance of penetration. The second is that we can't do a craniotomy on Monday, dose the patient, and do a craniotomy on Wednesday. So what we have to rely upon is matched tissue from the patient from the prior operation, which introduces new risk and new confounding factors, but frankly is the only practical way to do this unless you literally want to take a patient through 2 operations in sequence, which is really untenable if you're trying to do this in a high throughput way. The final is that, unlike other tumors like in the lung, the level of heterogeneity, and I'm not talking about genetic heterogeneity, I'm talking about histological heterogeneity for high-grade gliomas, for example, is much higher. So sampling one part of the tumor is not enough. So in our studies, we're sampling various regions of the tumor, enhancing and non-enhancing. It's relatively easy to get a drug into the enhancing component of the tumor. Gadolinium gets in, other things can get in if they're not huge and extremely hydrophobic. But that's not where the disease process challenge is. Most surgeons in this room can remove the enhancing component of a tumor. It's the non-enhancing infiltrative component that eludes our therapies. So this is what a basic phase 0 trial design looks like at our center. Patients get an experimental therapeutic, they wait several hours to days, they go to surgery, they get tissue sampled, then that's it. They move on to their other therapies. So this is a non-therapeutic study design, first and foremost. But it's designed to answer 2 questions. Did the drug penetrate, AKA pharmacokinetics? Did the drug modulate its target as it's supposed to, AKA pharmacodynamics? Many drugs that we design, and I'll give you a good example, EGFR inhibitors. We've been through dozens of iterations of EGFR inhibitors in human trials. Over 400 patients have been accrued to EGFR inhibitor trials, monotherapy, dual therapy. It never worked. It was finally after several hundred patients entered in trials that someone decided to ask the question, well, if we hit the target in a human glioblastoma, is it having the same downstream effect as it is in every animal model we tried? And the answer was no, because the EGFR pathway downstream in human GBM is not the same as it represents itself in a preclinical model. So the first drug that we chose to try this kind of approach at our center was a drug called AZD1775. The name's not important, but it's something that targets what's called the Wee1 kinase. So we know that there are different checkpoints glioma cells have. Glioma cells have to protect themselves too from abnormalities and DNA damage. Because most of them have p53 mutations, it pushes them towards one arm of these checkpoint streams, and that one arm has a downstream regulator called the Wee1 kinase. The discoverer of that won the Nobel Prize many years ago. It wasn't in glioblastoma, but it was actually in yeast. But everybody knew that linchpin was important for cancer cell regulation. So this drug was developed. It was first in class, meaning it's a new drug, new mechanism, hasn't been tried in brain tumor patients, period. We knew that there were some correlations that would suggest that Wee1 was a good target in glioblastoma. We don't have to get into the details of that, but one of the principles that we use in selecting drugs is really, has this been tried before? We need to convince ourselves that the target is viable and intellectually sound, but we also want to be trying new things for our patients. They don't need me-too strategies. The drug itself, if you dive into the details, was interesting. It has 2 different mechanisms of actually hurting cells, one directly through anti-tumor effects, one through the DNA damage pathways. We spend a lot of time analyzing the physicochemical pharmacokinetic properties of these drugs. It's difficult to look at a drug molecule and know whether it's going to penetrate the blood-brain barrier, but there are some features that make sense. The size of the drug, its partition coefficient, et cetera. All of these made sense, and the kinetics of the drug itself, meaning how long do you have to give the drug to actually see some sort of high level in the bloodstream, were favorable as well. So these things have to factor into designing the trials. Interestingly, this drug, when we looked at it, had been studied at Mayo Rochester by a friend of mine who does exceptional work in preclinical animal modeling, Jan Sarkaria. And what he showed was that the drug did not penetrate the brain tumors in these xenograft animals. And in fact, we see this with a lot of drugs in development, and usually the right conclusion here with conventional wisdom is, okay, this is not a drug to pursue in clinical trials. But the problem is that you just simply can't hang your hat on this preclinical data. And because of the data we had on how the drug was structured, et cetera, we thought otherwise. The trial itself was designed like this. There was an arm with a dose escalation. Patients had a glioblastoma. They had initial standard of care, surgery, radiation, Temodar. They had a recurrence. They were enrolled. And then at that time, they got a single dose of one of 4 or one of 3 doses of the drug. So just a single pill, which isn't going to harm anyone. They then went on to have an operation 8 hours after that pill was ingested, and then some blood sampling afterwards. Then there was another arm, another group of 8 patients, similar concept, except now the dose is fixed, and what differs is the timing of the operation after the dose. Some patients had surgery 2 hours after that pill, some patients 24 hours. So what this lets you do is it lets you take these tumor specimens and then ask these important questions. If you understand how WEE1 works in regulating cell cycle, then you should understand what should and shouldn't change as a consequence of effective inhibition of that target. And we can ask ourselves, is this actually happening? So there's 2 questions, again, in these trials, penetration and modulation. So drug penetration quantification is not an easy thing to do. Most centers don't have pharmacokinetic capabilities sufficient for CNS PK. Meaning it's not just about measuring the total concentration of a drug. That's not actually what's pharmacologically active. What's pharmacologically active is the free concentration of the drug. When you ingest a drug, it goes into your bloodstream, it binds to proteins, and if it remains bound to that protein, the likelihood of it getting into the blood-brain barrier, let alone the likelihood of it being pharmacologically effective, is very, very low. So the free drug in the blood and the interstitium is what matters. And so we develop methods to test that. The way you compare that measurement is to what some people today have defined, which is the IC50, the concentration of the drug required to kill half the cells in vitro. And so when you see free drug levels above that, then you know that you're getting reasonable penetration. And we did see that in this population and with this drug. And we could see it at different time points, at different doses, and you can quantify it. And what's called the unbound tumor to plasma ratio, that's the real metric of whether a drug is getting in or not. If it's greater than 1, that's a very good thing. If it's 3-fold or more, like it was in this drug, then it's exceptional. So this drug penetrated exceptionally well in humans, in contrast to the preclinical data that told us the exact opposite story. Now, the question of whether it modulates its target is a different question entirely, and you have to decide, depending on what the drug mechanism is doing, how you measure that. We chose several different measurements of DNA damage, measurements of cell cycle entry. You could see the concentrations and the fold changes here, and you can see that everything is above neutral, which means that there's a positive effect happening. And if you look at it by dose, you can kind of see a bell curve as well. So this kind of taught us a few things as our initial impression. The first is that there really is no substitute for human tissue data when we're doing drug development. There's only so much you can extrapolate from animal models, mathematical models, et cetera. The second is that, the patients that entered this trial were heroes to me because there was no value to them to do this. They went through this trial and took this drug, even though there was little risk, there was little upside for them as well. A single pill was not going to help them. So they're saints. But it does make it difficult to accrue, and it taught us that we needed to change the model to accommodate what patients really need. The third is that drug resistance really is one of the key questions when we're talking about targeting mechanisms. How is the tumor going to respond to that target? So you have to understand that longitudinally. You have to follow a tumor from time point to time point to see how it's evolving and responding. And finally, that bell curve that you saw in the pharmacodynamic effect, that's the reality of today's drugs. You know, the drugs that we all heard about 10, 20 years ago, those are chemotherapies. Those are toxic agents. And so the more you give, the more effective it is and the more toxic it is. Targeted therapies don't work that way. Targeted therapies have saturation points, those recursive pathways that will activate if you get too high of an inhibition rate. So the highest dose doesn't mean it's the best. And in fact, in many cases, the higher dose is less effective than an intermediate dose. So we adjusted our model to develop what we call phase 0-2 trials. And you know, we hear this term precision medicine a lot in our field. And I think for those of us that are really familiar with it, we know what it means. Precision medicine means you have a host of drug options, a host of different types of tumors, and you're mixing and matching based upon real logic and evidence. And that is not what we have in neuro-oncology. We have 1 or 2 drugs available to us and a variety of biomarkers that may or may not be relevant. I think the system we developed gets us a little closer to a real precision approach. So basically at our center, what happens is patients enter trial screening at the time of recurrence. They might get whole genome, whole exome, array CGH, whatever is necessary. They're then paired to the drug. Usually the combination of biomarkers we need to pair a patient to a drug is 3 or 4. So this is not a single biomarker type match. This is really us trying to predict what's truly going to interface with the drug mechanism. They get exposed to that drug, they go on to surgery, and at the time of surgery, all of those tissues are collected. Now, in the next 7 to 10 days, while the patient's recovering, they're getting their stitches out, they're coming out of the hospital, we're doing our analysis. By the time they see us in the post-op clinic, we know exactly how they've responded to these experimental agents. We can tell Mr. Jones, you know, you took 2 experimental agents at the time of surgery. I can tell you right now that both of them penetrated your tumor tissue in the non-enhancing and enhancing component. I can tell you that it modulated the targets in your tumor tissue in both cases. And so for that reason, I can confidently recommend that you continue on this study. For those of us treating patients, this is one of the hardest parts of treating our patients, is that we're putting them on clinical trials where they're taking a drug or therapy on faith. And we're asking them to wait 2, 3, 4 months with no evidence that for Mr. Jones there's anything's going to happen. Maybe the drug would have never going to get into Mr. Jones's tumor, but we won't know that for 3 months when we've lost a lot of time. For these trials, if that drug is not working for them, they know it before their staples are out. They can go on to any other trial without losing any time, and time is the one commodity that nobody has in this business. Now, once they graduate to that therapeutic dosing, now they're getting the drug therapeutically. There's side effects to these drugs, as with any experimental agent, but they're going to tolerate those side effects because they know that there's a reason they're getting exposed to them. It's not just for the greater good. So only patients that demonstrate both a PK response, meaning drug penetration, and pharmacodynamic effect get graduated. Everyone else gets pivoted to other clinical trials. And inevitably, for many of those patients, they have a recurrence. But when they have that recurrence, we then take them back to the operating room, as we often do for multiply recurrent patients. And so now for an individual patient, we have their individual journey. We have their tissue at initial diagnosis, we have it after a brief exposure to the experimental agent, and we have it after they recurred and evaded the experimental agents. And we can ask questions because we have DNA, we have RNA, we have protein from all of these time points for Mr. Jones. And we can say, what changed? How did it respond? What pathways were up or down? And what does this tell us in terms of what the next agent is we have to add on to this 2-drug cocktail, for example, to make it a 3-drug cocktail? To keep putting evolutionary pressure on this tumor until it runs out of options. And I'm always quick to remind people, these tumors are not magical, okay? They play by rules, and they don't have an infinite number of possibilities. So our job is to apply pressure until they run out of those possibilities and maybe not get cured, but become more indolent and chronic, more manageable by everything else we have at our disposal. So our first foray into this kind of approach was with another drug, another class of agent, a CDK4/6 inhibitor called Ribociclib. And I'll tell you, our center is pretty agnostic to whatever we're trying. We're not just focused on immunotherapy. We're not just focused on targeted therapy. We're not just focused on CAR T. Anything where there's a scientific rationale and an agent that hasn't been tried in human brain tumor patients, we're going to be open to doing it. Ribociclib was just one of them. And this is an agent that was developed for breast cancer. We know something about in what setting this agent works. It works when certain pathways are active. We also know that it needs the deletion of certain elements and certain sort of amplifications to have a chance at effect. So the trial works like this. The patient, again, has a glioblastoma. They go through standard of care. They have their recurrence. They go through trial screening. There's 3 different biomarkers for this trial that they have to have assessed. This is done through a combination of array CGH, immunohistochemistry, et cetera. Patients for this trial, because of the pharmacokinetic profile of the drug, got 5 days of the drug. Again, not enough to give them significant side effects, but enough so that we get enough in their body so that then they go to surgery. And at the time of surgery, they get tumor, enhancing, non-enhancing spinal fluid blood. This time, again, we varied the timing, 2 hours, 8 hours in some cases, 24 hours in some cases. But in all cases, we can again ask questions specific to how this drug is supposed to work. You know, if this drug works in breast cancer, for which it just got FDA approved, great. That doesn't mean it's going to work in glioblastoma. So we need to know that answer in these patients. We can then assess exactly what happens to the pharmacokinetic profile of the drug. We can ask ourselves, what happens in the spinal fluid? It's very difficult to know how to interpret spinal fluid levels. In some cases, you can have high spinal fluid levels that still doesn't get into the tumor, and vice versa. But nevertheless, in all cases here, the spinal fluid levels were quite high. And this, again, is unbound drug, okay? And here are the ratios of unbound drug and tumor to plasma and the enhancing component up to 40-fold. So that's exceptional. But it's the non-enhancing component that's the heavier lift. And again, up to 10 or 15-fold in the later time points, also exceptional. So overall, this drug is doing what it's supposed to be doing. It's getting there. But is it modulating its target? Well, you got to understand, again, how is it supposed to modulate its target? Typically, with cell cycle entry and CDK4/6, what happens is there's a complexing with cyclin D. There's a series of phosphorylation events. Some of the targets here include these proteins, RB, FOXM1, but ultimately, this leads to cell cycle entry. So that's what normally happens, okay? In the setting of an effective target inhibitor, if it's doing what it's supposed to do, then this phosphorylation event shouldn't happen. These downstream targets shouldn't be phosphorylated, and we shouldn't see cell cycle entry. So you can measure these. Now, I mentioned that one of the adaptations in neuro-oncology trials is that you can't have a crania on Monday and a crania on Wednesday. So we take the archival matched tissue for these specimens, and we first ask a question in another cohort of patients. The biomarkers that we're looking at, do they naturally drift from initial diagnosis to recurrence? And so we select biomarkers that won't do that, and then we ask ourselves in this cohort, or for this individual patient population, did we see an effect? And we did. The cell cycle inhibition appeared to be apparent. So patients are then selected to go on to phase 2. So we, I think, enrolled 12 patients overall in this study. 6 of them went on to phase 2. These are their swim lanes. Not surprisingly, you're not going to see a major survival benefit here with a monotherapy. Importantly, even though we went through several levels of rigor in trying to get homogeneity, there's still heterogeneity here. And this is the case with all trials, and we have to come to terms with the fact that even randomization doesn't necessarily give you homogeneity. But the point is that you get to see these patients' journeys, and when they recurred, we could ask ourselves, what is happening here? So I showed you this diagram of how CDK4/6 works. That's a simplification. It's a cartoon for our benefit. But we know that there are many recursive pathways that interact with this, one of them through PI3AKT, and you can measure that. And so what we saw was that in every patient that recurred, the mTOR activation spiked up, consistent with what we kind of understood for a regulation of this pathway. So again, very logically, this means that's the next step. And just this past month, in December, we started the next study. That study I just showed you, I reported at SNO in October. In December, we started accruing for the dual drug study, ribociclib plus an mTOR inhibitor. So what we learned is that following these tissues longitudinally gives you illumination of mechanisms of resistance. We know that single-agent targeted therapies have low therapeutic value, and so our emphasis has to be on building towards 2-3 drug combos. And everything we do at our center is intended towards that in the near term. But combined drug studies are complicated. most of the time when they're done with a conventional drug with a new drug. But how many times have we seen 2 or 3 new drugs put together? That's difficult. Why is that difficult? Number one, it's easier for a single company to do that. Well, most companies don't have complementary agents in their pipeline. So some of the best matches are between competing companies, and so that automatically that creates a level of difficulty. Number two, most companies and trials that do combine drug studies exclude brain tumor patients because they're trying to get FDA approval. Nobody wants to make it harder for themselves. Nobody wants to have toxicities that could be life-threatening and delay their application. And number three, it's actually quite difficult with the conventional rules of trialing to do dose finding with this, because the way you typically do 2 new drugs is you start them both off at super low levels and you ratchet them up 1, 1, 1, 1, 1. Well, that's not really a therapeutic study. Those patients in that dose escalation study are maybe not going to derive any benefit from that. But when we looked at the whole history of oncology targeted therapies, for example, for combo drug studies with 2 new drugs, what you learn is that at the end of the day, after years of investment and ratcheting up doses from super low to something therapeutic, they almost always end up at the MTD or the maximally tolerated dose for the individual agents. So all of this ratcheting up was really unnecessary. You could have started at the high levels already. Number two, you don't have to keep pushing dose levels until toxicity, right? That's the concept of a phase 1 study. How much drug can I give a patient before they're so sick that they can't get the drug anymore? But that's a very anachronistic way of looking at chemotherapies. We're looking for molecular effect, not toxicity. And in fact, if you look at the data, the intermediate doses of many drugs are just as effective as the high doses where they're getting toxicity. So we developed a study design that we call phase 0, 1, 2. And the basic concept of it is that you start already at 80% of the MTD for any individual new drug. You combine them at that point, and then you ask a question. Are they sick? No, that's not the question you ask. The question you ask is, have we reached a floor of biological effect? Have we gotten, with 80% of each drug, at least the minimum level of drug penetration and the minimum level of drug effect? If the answer is yes, then that patient moves on to the therapeutic dosing. And then within that patient, you dose escalate. See, at that point, the patient's already covered. They're already getting clinical benefit potentially from whatever you're giving them. And then you can see what makes them sick. So I'm getting near the end of the talk. I'll just end with a couple slides giving you sort of our overall approach to selecting drugs for drug development program. The first is that everything has to be new, but we're not in this to build things from the ground up. There are plenty of good drugs, good mechanisms that haven't been tried in this patient population. Repositioning, repurposing these drugs, early phase drugs, not FDA-approved drugs, is a viable strategy. The second is that we really shouldn't be in the business of trying to predict what's going to work. We've been very, very ineffective at that. So we have to be open to all solutions. The third is that you really can't do these kinds of trials without biological context. It means that if you give a drug and patients do well, great, but why? Unless you have a real evidence of drug effect, it's hard to interpret that data. And finally, every trial, from my perspective, needs to either have the intention of getting you to a dual or 3 or 4 drug cocktail, or it should just start with that. So at our center, I think the guiding principles are, one, preclinical data is very, very useful, but to a point. And you have to be quick to make the jump to the clinical platform. Number two, you can ask these preclinical questions in humans and get better and more reliable answers. Number three, any clinical result needs to have biological context. I'm happy to have a trial where you have a p-value and a significant survival benefit, but more importantly is why, and is the effect of the drug what is actually happening. And finally, we basically don't choose an agent for our trial portfolio unless we already know 2 or 3 combined agent possibilities that we're also going to assess at the same time. Everything is moving towards cocktails. So I'll finally just end with a picture of our organizational chart and the many scientists and non-scientists that are part of the team. Our legal department is probably as important as our pharmacokinetics department in terms of moving things from the left to the right and pushing us through. And obviously, the patients that are putting themselves out there for this because they want to have something new. They want to have a possibility of a outcome that's different from everything else they've seen, which in my book means they need to try something different from what everyone else has had. And of course, I'll thank you for your time and attention. Questions?
I have a question. So I have a question.
Shoot.
And my question is, you've identified a way of, again, I don't want to use the word bypass, but maybe skirting around some of the limitations that are inherent in the system. And you've talked about some of the financial pressures that have driven the course of development that we have today. Is there a breaking point where we will be able to say, this system doesn't work anymore, right? This system for developing new drugs and treatments, at least for this population, is broken. And to me, it's similar to some of the areas in research where we've said a randomized controlled trial is great, but doesn't apply here, right? We need some, whether it's a prospective registry or some other mechanism to prove what we're going to do, because we can't conscionably do a randomized controlled trial on this population. And what is the breaking point where we say we can't conscionably adhere to the existing paradigm for drug development and make any progress for a population that has no alternative?
I think everyone's heart is in the right place, but what we're seeing are things moving forward in drug development that maybe shouldn't, and other things not moving forward that should. So a concrete example, in the last 6 months, the EORTC, which is a huge European consortium, announced a new phase 3 study for a new targeted inhibitor. 781 patients will be committed to this set of trials, and I won't go into the details of it. But if you look at the drug that they chose, and you ask yourselves, is there sufficient evidence to justify this? How excited should we be about this drug? And you ask the people that are involved in, the taste making for this particular decision, the answer is that the evidence is thin. Some of these trials get greenlit for a variety of reasons other than the logical ones. This is the world we live in. On the other hand, if you look at glioblastoma, and if you were all honest with ourselves, the TCGA data set subdivided it a several-fold, but it's probably several orders of magnitude more divided than what we even know. I think a good example of where our field is headed is, for example, you've heard of Neuro-Oncology and the pan-track inhibitors that they and their competitor have put together. This is the first FDA-approved drug that was approved without an actual histological indication. It was approved for a pathway, irrespective of the disease it's in. And in fact, there's only been a handful of patients in the world treated with this drug, and glioblastoma, literally probably less than 10, because it's less than 1% of all glioblastomas. But in one of the patients that did get treated, and in some of that handful, they've had remarkable effects. But that's not going to be applicable to the general population. So this is where we're headed. This disease process has to be individualized well beyond the point of what we're comfortable with in oncology. It may not be something that lends itself to large-scale, 700-patient, 5-year, 7, 8, 9-figure clinical trials. So for us, the journey begins individually. We're looking for individual solutions for individual patients. Ultimately, that will inform larger-scale decision-making about how to treat these patients. But we think the solution is going to be percentage by percentage of this population. And hopefully in the future, while we're all still here, patients with a glioblastoma are going to come in. They're going to have 4, 5, 6 biomarkers that identify some sort of signature, that identifies some sort of subset they're in. And we're going to have a combination of drugs and therapies that are going to target that subset. And it's going to be a few percentage points of the overall. And we're just going to have to go slice by slice through this pie. I think that's the way to parse this out.